Which of the following is not required for any of the techniques of DNA fingerprinting available at present? Option A, polymerase chain reaction, that is PCR. PCR is a crucial technique used in DNA fingerprinting to amplify the small amounts of DNA sample, allowing the analysis of specific DNA regions that may differ between individuals. So, option A is an incorrect answer. Option B, zinc finger analysis. Zinc finger analysis is not a commonly used method for DNA fingerprinting. They are protein motifs that bind to DNA and can be used in gene regulation or gene editing technologies. So that means option B is the correct answer. It is zinc finger analysis. They are not part of the standard methods for DNA fingerprinting. So option B is the correct answer. Option C, restriction enzymes. Restriction enzymes are used in DNA fingerprinting techniques, that is to cut DNA at specific sequences, which helps in creating a patterns of DNA fragments for comparison. So option C is also incorrect. Option D, DNA-DNA hybridization. DNA-DNA hybridization is used in DNA fingerprinting methods like Southern blotting. So, to identify specific DNA sequences by using labeled probes that find to complementary sequences. So, that means option D is also incorrect. The correct answer is zinc finger analysis.